Hello, everyone. I'm Sean Gibbons, a faculty member and microbiome researcher at the Institute for Systems Biology, or ISB, in Seattle, Washington. ISB, if you haven't heard of us, is a nonprofit biomedical research institute. We're an affiliate of the Providence Health System. And we focus on the most pressing issues in human health. Systems biology enables ISB scientists to better understand the human microbiome, as you'll learn about today, but also cancer, aging, brain health, chronic and infectious diseases, and much more. ISB also focuses on K through 12 STEM education and beyond, as you'll see here today, in particular by addressing inclusion and equity for all scientists, including historically underrepresented groups in science. You can learn more about ISB by visiting our website, isbscience.org. As a nonprofit research organization, we rely on the generous support of our donors. ISB puts on a number of virtual events every year, and we have a couple of upcoming ones in November that I'll tell you about, just to keep on your radar. On the evening of November 13th, we're hosting an in-person event called Ignite STEM that celebrates the impact of ISB's education team and that it's had on teachers and educators in the US. On November 21st, at noon Pacific time, we will host our final research roundtable. It is ISB Associate of the Year, it's ISB Associate Professor Dr. Wei Wei. And he's going to talk about a technique that he's developed, his lab has developed, that will provide real-time information to physicians to inform precision cancer treatment. And you can find those events on our website under the Events tab. We put these events on at no cost to registrants. So, if you have support to give, we'd greatly appreciate it at isbscience.org/donate. You can find a link at the bottom of our course website as well. We hope that everyone in the US had a great Indigenous Peoples Day back on the 14th, back on Monday. We acknowledge that we're here in Seattle on the traditional land of the Coastal Salish, including the Duwamish people, past and present, and we honor with gratitude the land and the people. Okay, so before we begin, I need to express our gratitude to our generous sponsors who allow us to put on these events free of charge to our registrants. First off is Applied Microbiology International. And not only did they provide financial support to this event, they also are providing free memberships to all registrants. So please take advantage of that opportunity. We also would like to thank the Environmental Health and Microbiome Research Center, or the Embrace Center at the University of Washington, and Science in Seattle, the website for supporting this event. Because of their support, we're able to provide this course and the following symposium to, I guess over the past 5 years, it's been about 6,000 people from about 80 different countries around the world. To all of you tuning in from around the world, we're grateful for your time today. We're excited for this opportunity to help train the next generation of microbiome researchers, or micronauts. If you're on social media, please remember to share your experiences throughout the 2024 ISB Microbiome Series using the hashtag #ISBMicro24, and that'll be on the slides throughout the Zoom. And if you want to tag ISB itself on X or Twitter, we're at ISBSCI or ISBSci. So right now, we're gonna launch a poll to kind of check in on where everybody's zooming in from around the world. So please participate in that and let us know where you're zooming from today. And while that's going, I'll give a little bit of background on today's course. So this is very similar to the structure of this course from the past few years, if you've joined us before. The first day is really focusing on 16S amplicon sequencing data analysis. And the overall theme of the course this year is the gut-brain axis, exploring how the microbes in our gut may be affecting the central nervous system. And our instructor today, Alyssa Eastonon, has put together a really great curriculum for learning how to use 16S amplicon sequencing data to tell us something about how microbiomes vary between healthy patients and patients with Parkinson's disease. So I'll give a quick intro to all the team. So of course, I just said Alyssa Easton, a graduate student in the lab, is our instructor for day one this year in the molecular engineering graduate program at the University of Washington. We also have TAs who are on the course Slack, who will be helping people troubleshoot and answer questions. So our TAs are Jacob Cavon, Noah Rappaport, Alex Carr, Crystal Perez, Carl Geiser, and Nick Quinn Bowman. Our admin support today, the folks who are behind the scenes making this all happen, are, of course, Dominic Lewis, who's our coordinator, Allison Kudla, Joe Meister, Audrey Hubbard, Erica Krager, Ray Rosario, and Thea Swanson. Thanks so much to all those people for helping put this together. All right, so hopefully you've had time to respond to the poll. Let's now quickly look at those results. Now, let's see, where did it go? Oh, here we go. Okay, so now I see them. So I wasn't too ambitious with thinking about what this would show me, because we are in the Pacific time zone here in the US, and I realize that it's very hard for people from Asia or Africa or other continents to join us in live for these events. That is why everything we're doing today is being recorded and live streamed and will be available on YouTube, in addition to all the materials being available in perpetuity after the live event is finished. So those of you who are still asleep, don't worry, you'll have access to everything. But thanks to all of you who have joined us. It looks like about 60% of folks are coming from North America, which isn't too surprising. But we have 23% of our registrants currently with us from Europe, 10% coming in from Asia. We have 3% from Africa and 4% from South America. Sadly, I don't see anybody from Australia, but of course, that is a very difficult time zone to zoom in from live. Thanks to everybody for joining us live. It's nice to have you here with us. And the one benefit of being with us live is you really do get that live interaction with our TAs on the Slack channel. So if you have questions, you can engage with us and get those answered. Okay, so if you have issues, like I said, you can visit the Get Help channel on the Slack. You've already received a link in the email to join the Slack. But we'll post links to those things on the Zoom and other places to make sure you have access to all of that. To ensure that everyone can enjoy the course in a safe and harassment-free environment, we provide a code of conduct, which can be found pinned to the general channel of the Slack. If we observe behaviors that violate this code, we'll warn perpetrators. We reserve the right to remove problem users from the Slack account if their behavior persists. This has never been a problem in the past. Everybody's always very well behaved, so thank you. Okay, so that is the end of my prepared comments. I will quickly say at the end here that we started these events during the pandemic, right, as a way to allow access to training, bioinformatic training for microbiome data analysis to a wide variety of people virtually, so that they didn't have to go anywhere in person, and make it free. So it's easy, there's a low barrier essentially to having access. And we've done it now for 5 years. This is our 5th year. And I think it was well appreciated, and we've loved doing it, and we could keep doing it again and again. But I think we'll probably call it a day after this year. This might be our last virtual series. We'll probably continue to do our symposium, but maybe not the course. It may get rebooted in the future, and we may try to do something else new and creative. But that's just to say, you're at a very special time where this is perhaps the last one of these that we'll be throwing. And we're going out with a bang with this gut-brain axis theme. So we appreciate all of our registrants over the years, and we hope you really enjoy these next few days. Okay, so now I think I will stop. We are going to officially start the course at 9:30 Pacific time, so in a little over 15 minutes from now. So Alyssa Easton, who is our instructor for today, will be here in about 15 minutes. But in the meantime, we're going to take a quick break and kind of adjust and reshuffle and get everybody situated. So we'll see you back here in a few minutes. Grab a cup of coffee or whatever you want to drink, because it's probably the afternoon or whatever time it is where you are, and enjoy the day. We'll see you soon.
Welcome, everyone, to the first day of the ISB Microbiome Series. As Sean mentioned, my name is Alyssa Eastonon, and I'm a graduate student in the Gibbons Lab. I'll be your instructor for today's part of the course. I'm so glad you could join us today, and I hope you'll also stay for a wonderful talk later, as well as for the next 2 days of the series, where we'll have another part of the course tomorrow, and we'll finish up on Friday with a symposium featuring several exciting talks. So let's begin. The first thing we're going to need is the slides. Oh, here, wait. There we go. So the slides that I'm sharing with you right now are available at this link. They are also linked on the course website and in the email you should have received yesterday. The TAs will also post a link on Slack soon if they haven't already. And I encourage you to open up the slides and go back through them later if you need a refresher. But yeah, follow along. So the organization of this course is going to be the presentation slides that we're looking at right now. They're going to contain the theoretical background and logic behind some of the methods we'll cover today, with explanations and links to additional resources. And second, we have the interactive course notebook, which will be opening in Google Colab. And this is where we will handle the technical aspects of the course. So you'll follow along as we process and analyze data in real time, generating visualizations that help us learn about the microbiome and this particular data set that we'll be analyzing. And then lastly, we have Slack, which is our chat. And the link to the Slack channel is in the introductory email. And I believe also on the course website. This is a great resource for asking questions or getting technical support. And while the Zoom chat won't be monitored as actively, the Slack channel will be monitored, and TAs will be answering questions as they pop up. So, oops, here we go. Let's switch to the notebook. So if you have the slides open, you can click this link to open up the notebook. And if you don't yet, the notebook is also available in a link on the course page and also on the GitHub. Okay, so now we're looking at this notebook. I'll give you a moment to open it up. And once it's open, you should see something like this. This is the Google Colab. And before we begin, the first thing you should do is make a copy of this in your own Google Drive so that you can save your outputs and access them later. So I will also do this. I will show you. If you do File, save a copy in Drive. And we are saving a copy. It might prompt you to log into your Google Drive. And okay. And then you can rename it whatever you want. So now that you've saved a copy, let's take a moment to understand what we're looking at. The notebook is a tool that allows you to run through the data analysis in real time. It's divided into different cells, some of which contain text, like this introduction here, where I have links, and then some of the cells are going to contain code that we can run. So the code cells are slightly darker, and they have a play button. When you put your little pointer over them, you can press play. You can also play them by hitting, I believe, Shift Return or Shift Enter. So go ahead and run the first cell, I will do so with you. We are cloning the materials directory from our GitHub into this local environment so that we can work with the data. Okay. Once this is done, we're going to look at this folder icon on the left of the screen. And it should contain a folder called materials. You can open it and check it out. Okay. So, yeah. Then we can navigate to the directory in our notebook so that we're running the code from within this folder. And notice that we use a division symbol instead of an exclamation mark. And that's because we want to make this change permanent in our Colab notebook. And we're almost ready to get started, but we need to first install the tool that we're going to be using today, which is called Qiime. So run this next cell and start it now because it's going to take a little bit to download. And there will be little updates here as you're downloading it. And while this is working, it might take about 10 to 15 minutes. So we're gonna let it run and go back to the slides. This installation is only in your Google Colab environment, so nothing will be installed on your local machine. Okay, let us switch back to the slides. Oops. Here we go. All right. So, the gut microbiome, what we're going to be studying today. Let's dive into some background on it. By being here today, I assume that you're somewhat familiar with the gut microbiome, but let's have a quick refresher. So the gut microbiome refers to the community of 30 to 40 trillion bacterial cells that reside in the large intestine. These bacteria play critical roles in our health. They live primarily in the mucus layer of the large intestine, and the composition of this community can vary significantly between individuals. So each of us has a unique set of microbes in our gut. And these microbes do a lot of things for us. One of their primary functions is aiding in digestion. For example, they can help break down resistant starches or cellulose that our bodies can't digest on their own. The bacteria metabolize these compounds, producing metabolites that re-enter the body and impact our health in a lot of different ways. This could range from reducing inflammation to potentially increasing the risk of chronic diseases like cardiovascular disease. And even beyond digestion, the gut microbiome plays a big role in training our immune system, determining our susceptibility to pathogenic invasion, and even modulating mood and behavior through the gut-brain axis, which is the theme of today. Let us go to the next slide. And here I've drawn out a pretty complicated figure, but we're gonna talk through it and hopefully it'll get you excited about what we're gonna do today. So I kind of like to think about gut-brain signaling in 3 different categories. The first category is a direct metabolite transfer. So certain microbial metabolites can diffuse through the intestinal epithelium where they are taken up by the portal vein. And then the portal vein takes metabolites directly to the liver where they're filtered and modified. And then some metabolites enter systemic circulation. And although we're up here now. Although the blood-brain barrier is highly regulated, certain metabolites like short-chain fatty acids and amino acids can actually diffuse right through the blood-brain barrier into the brain, where they might act as signaling molecules immediately or as precursors to signaling molecules like neurotransmitters. The second category I like to think about is immune-mediated gut-brain interactions. So the microbiota is in constant communication with the host immune system, and it shapes both the body's balance of pro and anti-inflammatory processes, as well as the integrity of the epithelial barrier here. And in the case of chronic low-grade inflammation, we see the barrier degrade and more bacterial metabolites are able to cross the barrier. So inflammation in the body over time can also end up chipping away at the blood-brain barrier and sort of increase the ways that microbes might be interacting with your brain. The last category I think about is direct neuronal signaling. So the enteric nervous system intricately connects the gut to the brain through both the vagus nerve and the spinal cord. It sends and receives signals from the gut epithelial tissue, from cells that are our cells. And then in some cases, it sends or receives signals from the microbes, molecules that have diffused into host tissue. I always like to talk about this paper that showed a microbial fatty acid amide that activated endocannabinoid receptors on gut spinal afferent neurons. And that signal directly from microbes caused or controlled a dopamine circuit in the brains of mice that caused a runner's high. So of course, these categories aren't comprehensive. There's so many other body systems and cell types that gut-brain signals could be acting through. But I hope it's gotten you excited to look at some data today. Okay, let's go. So today and tomorrow, we're going to be analyzing a case-controlled data set from a study of Parkinson's disease. And there are a few reasons why a PD is particularly interesting to gut microbiome researchers. But the main one is that in many cases, PD seems to begin in the gut. So years before motor symptoms develop, PD patients develop GI symptoms like constipation, increased gut permeability, and inflammation. And we know that these things are generally not good for you. So the current question is, are early changes in the gut contributing to the development of Parkinson's? And how might the microbiome be involved in this? If you look at the link here, I've linked a meta-analysis from 2021 that included many Parkinson's disease microbiome correlation studies, and they found some common threads related to gut inflammation. But the answer to our question is still pretty uncertain. And we're not going to answer the question for you today, unfortunately, but we are going to look at one of the data sets that this metadata or this meta-analysis included. It is from, oops, sorry. It's from Hill-Burns' 2017 study that included over 300 participants, and they collected 16S, or they collected stool samples, processed them, extracted the bacterial DNA, and did 16S rRNA amplicon sequencing of the stool samples. This data set is also interesting to me particularly because it includes demographic information, health history, and medication use, which is actually a very important confounder in a lot of observational studies of the microbiome. And this study was able to identify small but significant independent effects of Parkinson's disease and Parkinson's disease drugs on the microbiome composition. So today, we're going to be looking at a very small subset of the original data, just 10 participants for computer processing reasons. but this might be a spoiler alert. I might have processed the entire data set for you, and we will actually be able to look at the whole data set things in the end of the notebook. Okay. So, measuring your microbes. Given the microbiome's influence on health, you might wonder, how do we know what is in someone's microbiome? And how do we measure the microbes that are present in one sample? So this is where sequencing comes in. We take a fecal sample, extract the DNA, and sequence it to identify the microbes and their relative abundances. However, sequencing raises several challenges, including processing the data and analyzing it meaningfully, because, as we'll talk about later, sequencing is kind of like taking a random sample from a sample. Okay, go here. So what we're going to do today is, starting out with raw sequencing data, we're going to trim and filter the reads before we infer amplicon sequence variants, or ASVs, which are the most likely set of original sequences present in the sample. With those ASVs, we can calculate alpha and beta diversity metrics. That's not the next thing. That's actually, yeah, something else. Yeah, but okay, so we can also move forward with the ASVs and then assign taxonomy based on a database of reference genomes. And then we can normalize the data and actually do some differential abundance analysis. Okay. Oops. So the tool that we're going to use to do all of this is called Qiime. And it's now on the second version. But Qiime was originally developed around 2010 by Greg Caparezo and Rob Knight during the Human Microbiome Project. So it streamlines the process of transforming raw sequencing data into meaningful outputs. So with Qiime 2, which was released in 2016, there's a strong emphasis on transparency in the analysis process to allow us to trace exactly how the raw data is processed. And this is so important for having reproducible results to our data. More information about Qiime is at these links. And what Qiime really is a tool to process the sequencing data, generate intermediate outputs, and create visualizations to analyze microbial communities. You'll most often see Qiime used via the command line, as Chris Diener is doing here in this little GIF. But today we're going to be working within the Colab notebook. So it looks a little bit different. We have the exclamation point in front of it, but the commands that we're using for Qiime are the same. So Qiime 2 is organized around what we call artifacts and visualizations. And these are different data types that Qiime uses. So an artifact is going to be a QZA file, while a visualization is QZV. And artifacts are intermediate data files generated during the analysis, while visualizations are endpoints specifically for us to put into a browser visualization tool that Qiime has to look at. So for example, we start with the raw sequences. We can put them into Qiime as an artifact. These raw sequences feed into various methods. The method is represented by yellow rectangles, like demultiplexing, denoising, and taxonomy classification. And then each method produces new artifacts that go on to the next process. Yeah, and then the blue ones are the visualizations we can do to look at every step of the process, make sure we're doing this data analysis correctly. Yeah, and just a note, once a visualization is created, it can't be used as an input to another Qiime method. So you can't put a QZV file and do taxonomy classification on it. It has to be a QZA. Yeah. So let's make our first artifact. Let's switch back to the notebook and get started with that. Okay. So we will, oops, do a little escape and get back to the notebook. Oops, I'm just going to close out of that. Qiime should be done installing. And our first cell is here. Oh, yeah. Okay, so first, we're going to make an artifact, but first we need to, let's check out the actual raw data that we're going to be using. So if you open up the data folder in the materials directory, you're going to see these are our FASTQ sequencing files. And you also see something called the manifest and metadata. These are tab separated value files, or basically tables. So let's look at what these different files are and why we need them to import our data into Qiime. So we use pandas to make this into a nice table for us. And this is our manifest. So the manifest file maps sample names to the location in our directory of the FASTQ files. So we use the manifest to tell Qiime where to find the raw sequencing files. And the metadata file, which is the next cell we can run, that has all the additional information that's not included in the sequencing data. So it has our sample ID, so we can match it up with the sequencing information, but it also has things like whether this person has Parkinson's disease, and other covariates like sex, age, BMI, and some medication usage and diet information. So let us go down to this section called FASTQ to artifact. So to use this sequencing data, we need to turn the FASTQ files containing our data into Qiime artifacts. And we can do that with this thing called Qiime Tools is the plugin. And then import is the action that we're going to use. So go ahead and run this. It should take about 20 to 30 seconds. And what this is telling us is, or we're telling Qiime that the type of sequencing that we have is this kind of sequencing sample data. And then we also have the input path. So that is going to be our manifest or our mapping file. We have the output. We're going to output the Qiime artifact to something called sequences. And then our input format tells it Qiime what type of sequencing this is. So you'll see it says single end FASTQ. That is because this was single end sequencing, and the file format is FASTQ, which is a sequencing file that also contains quality information, and we'll talk about that in a second. Okay, so just going over this again, this is what the Qiime command format looks like. So we say Qiime plugin, action, and then arguments. And the arguments are usually going to follow the order of input, output, some different parameters. These can be kind of variable, and then metadata, which is so that we can always have our metadata mapped back to our samples. And then before we do anything with the sequencing data, we want to look at the quality of it, because as we'll talk about, there are going to be some issues with it. So we can turn this artifact sequences.qza into a visualization using the Qiime plugin dmux, which is short for demultiplex, and then the action summarize. And this will take a few seconds. But once the cell finish is running, we're going to open it at the Qiime viewing site, which is called view.qiime2.org. There's a link right here. And so I will open it up. You're going to get a redirect notice, but that is okay. Okay, and you'll see the Qiime2 view site looks a little like this. They have like little examples. If you don't have your own data to use, you can still explore how this works. But you can basically drag and drop or click and select a file to open in Qiime2 viewer. So we have now saved this visualization. So let's look. We're in the materials folder. I'm going to close the data folder, and then it should be in here. Here we go. So qualities.qzv. We have to download this file. There it is. And then we can just drag and drop it into Qiime. So let's check it out. Here we go. So we can see here. Oops. Yeah, okay. So this is a histogram of the number of sequences. And we also have, oh yeah, so since this is single end sequencing, we only have forward reads. But if we did paired end, we would also have reverse reads. And we want sort of a certain number of reads per sample. And what this cutoff is kind of depends, but for today, I set a cutoff of around 5,000 reads. And then we can also look at the interactive quality plot. And this is going to show us the quality, the average quality over the length of a read. So the unit is in base pairs. So what you will see is a quality score. And what the quality score actually means is that a quality score of 30 corresponds to a 1% error rate. So that is here. And then a quality score of 40 corresponds to a 0.1 or 0.01. I think a 0.1% error rate. So based on this plot, we'll decide where to truncate our reads. We're going to kind of cut off the reads at a certain point because we usually don't, or we're going to see issues with quality in the beginning of our reads due to the actual physical process of amplicon sequencing. So where do you think we should truncate these reads? I guess we don't have anything that's below 1% error. So that's kind of good. Let's see. Okay. So today we're going to just be using a truncation length of 150. So we're going to keep this whole thing because the quality does seem to be pretty good. But this is, something that you can change, and you might have to if the quality score is very low in the beginning of your read. Okay, so now let us, oh yeah, okay. Head back to the CoLab notebook, and we're going to start this process, the next process in the data cleaning, which is called denoising. So go ahead and run this cell now, because it's going to take a little bit. And we'll head back to the presentation slides to talk about what the cell is doing. And here we go. So, oops. So to understand why we're doing all this data cleaning, we need to talk about the errors that are generated by sequencing itself. And to do that, we need to know what type of sequencing we've done. So today, we know we're working with amplicon sequencing data, which is after we extract DNA from our samples, we focus on a specific gene called a marker gene, which we amplify via PCR. And then we can sequence just this gene. And in shotgun metagenomics, which is not what we're working with today, we actually are looking at little pieces of the whole genome. And then we have to kind of map them back together from all of these little reads, but from lots of different organisms. So we have to match them. So it's a different computational process, but let's talk a little bit more about 16S. Okay, so this is the 16S gene. And we use it because it is in all bacteria, so it's universal, but it also contains interspersed regions that are conserved, which are good for our primers to latch onto. So they're not going to be changed in different bacterial species, but they also have hypervariable regions, like if you're familiar with antibodies. These variable regions have a lot of phylogenetic heterogeneity. So these allow us to distinguish between different bacterial taxa. And our data specifically used this V4 region, but there are other variable regions that one might use in 16S amplicon sequencing. So the V4 specific primers we used in this study were F515 and R806. And the F stands for forward and the R stands for reverse. So I have a question for you to maybe answer in the Slack. How long is the amplified fragment? And this might be a trick question. How long are the reads?
Yeah.
No? Oops.
People, one saying 290, one saying 300. Yeah.
Well, the amplified fragment, okay, I guess I'll just give you the answer. But thank you for those who did answer in the Slack. The amplified fragment is going to be the length between the forward and reverse primer ends. So that's going to be 806, which is the base pair number at minus 515. And this actually has nothing to do with, I guess this figure is kind of wrong, because the amplified fragment here is actually longer than the read length we used during sequencing. They're different processes. So our sequencing read length was only 150, even though our amplified fragment was this long. So now with either method of sequencing, whether amplicon or metagenomics, we're going to have the issue where we talked about read quality isn't going to be great at the beginning of the read. And that has to do with the physical process of next generation sequencing methods, which we're not going to really get into today. But we are going to talk a little bit about why sequencing, or why 16S data specifically has some errors that happen during the PCR amplification step. So this little fragment here is called a chimera, and it is formed, it's like a hybrid between one piece of DNA that was in your sample and another. So during PCR, fragments from different DNA molecules might join together incorrectly after the extension step if it doesn't finish completely. So 16S sequencing is going to have artifacts in it from these chimeras. And we want to remove them because this obviously is not an actual or a true sequence that was in our sample. We also have, next generation sequencing has a certain error rate, which is pretty low, but when you're using, or when you're doing many reads, the number of errors is non-negligible. So we need to be able to tell if we have these three reads and they each have like a different base pair here, and but that is an error. We need to be able to tell that that's an error and know that these are actually coming from just one original sequence. And that is where this plugin that we just ran called Data2 is going to come in. So denoising, the first thing it does is it trims the reads at whatever length we specify. So we specified 150. And then it also will remove the reads that are of a certain, or below a certain quality threshold entirely. It'll remove reads that have bases that aren't assigned. So instead of A, C, T, or G, it'll be N, that the sequencer couldn't decide. And we also remove sequences that were added to aid in the sequencing process. So we only have the sequences from our sample. Then we also, steps 3 is remove the chimeras, like we talked about, while also finding the most likely set of original sequences, or amplicon sequence variants, ASVs, like we talked about in the last slide. And then we have to count them per sample. And how we do this is oops, here we go. Okay. So how we do this is with an algorithm called expectation maximization, which actually simultaneously assigns the ASVs while it's modeling error. And this is an improvement from the previous method used, which was what we called OTUs, if you've worked with sequencing data before. That method used kind of an arbitrary similarity threshold, while this one decides similarity based on iteration. So this algorithm within Dada2 creates our ASVs, which are going to be smaller, or a smaller amount of original sequences than we started with, because we have controlled for these errors. So there we go. We'll have 3 outputs from this. We're going to have a feature table containing the abundances of each ASV in each sample. We're going to have a file of representative sequences, which are the unique ASVs, all of them kind of in one file. And then we're also going to have the denoising statistics, which summarize how many sequences were filtered out or retained during the denoising process. So let's switch back to the notebook, and we will look at how that process went. So my Dada2 is done running, but if yours has not finished or if it has failed, you can stop your cell if you click on this again, and you can copy the results from our treasure chest, which is where we've put all the files that we're going to generate here. This will only run if your previous step didn't work, so it won't hurt to run it. Okay, so then we should have a folder called data in our materials folder. So if we open this up, we're going to see those things I just talked about. And what we're going to use is the denoising stats Qiime artifact. So we want to look at this in a visualization. So we can use the plugin metadata and the action tabulate to make the denoising stats into a table that is a QZV that we can look at in the Qiime2 viewer. So this should take a second to run. Okay, so now this should have appeared in your Dada folder. We're going to download it. Okay, and if we go back to Qiime2, we can get it in here. Okay, so all 10 samples are there. These are our input read numbers, and then after we filter, here's our like endpoint read numbers. So the goal is we want a certain number of filtered reads, because we want to have enough reads to have sequencing depth to tell apart different taxons, or yeah, we want enough depth to like measure a certain amount of the microbiome. But here we have, this will tell us like how many chimeras were trimmed. So the amount that were non-chimeric are here, and it's pretty normal for your number or your percentage of chimeric values to be between 5 and 25%. So all of these percent non-chimeric being between 75 to 90% is pretty normal. And that means looks like this process went well and we can move forward. So let's go to the notebook. And we can go ahead and let's just start running the next cell before we head back to the slides. And I'm going to talk about phylogenetics and diversity. But sometimes this tree can take a little bit to run. Okay. So full screen. We are running the phylogeny first because there are some diversity metrics that we want to calculate that are based in phylogeny, which we'll get to in a second. But to start, we have two main types of diversity. And diversity is a crucial aspect of microbiome analysis. And we typically measure it in either alpha diversity, which is the diversity within a sample, or beta diversity, which is the distance or differences between samples. So let's talk a little bit more about what I mean by that. Alpha diversity measures the diversity within a single sample. And these components of this metric are things like richness. So how many total taxa do we observe? Or evenness, how evenly are these abundances distributed across the taxa within a sample? And then some certain metrics are going to use different combinations of both richness and evenness. So today we're going to use the Shannon index, which combines these. And the Shannon index basically answers the question, how surprised would we be to pull a specific taxon from the community at random? And the higher the diversity of a sample, the more surprised we'd be. So then we also have beta diversity, which measures the diversity between different samples. So it answers the question, how different are two or more samples from one another? And there are two types, unweighted and weighted. So the unweighted beta diversity is how many taxa are shared between samples. And this is measured commonly by things like the Jacob index or unweighted unifrac. And then the weighted measurement is, okay, among these shared taxa, do the ones that are shared have similar abundances? And metrics that take this into account are things like Bray-Curtis distance or weighted unifrac. And what we are going to be looking at a little bit today is called unifrac beta diversity. And in this beta diversity metric, the distance that we're using is based in phylogeny. So we're asking the question, do samples share genetically similar taxa? Where the unifrac distance is actually the unshared branch length. So our distance metric is going to be, if we have two samples, it's the amount of total blue or green, teal branch divided by the total branch. So that is teal plus black. So this sample here has pretty similar things. So we are going to, so its unshared branch length is going to be shorter. And then this one has these taxa that are really far apart from each other. And they are going to have a higher diversity metric. So we also talked a little bit about the weightedness of diversity. So the weighted unifrac scales the branches by the abundance of the taxa, so that the presence of one really distant member, for example, doesn't skew diversity towards being really diverse just because you have one taxon that's never been seen before and is really different. Okay, so what we're running right now is we're building a phylogenetic tree. And in order, or this tree is going to cluster the taxa based on their genetic similarity. So the shorter the branches are between two taxa, the more closely related they are. So let's switch back to the notebook and check on the tree, which we are using the ASVs we got earlier to build. Okay. Oops. Here. So my tree has saved to a new directory called tree within the materials folder. If we open this up, we can see our outputs, tree.qza and rooted tree.qza. And if we want to look at these, we have to create a visualization again. So we'll run this cell, and we're using a plugin called Empress that is specifically for making visualizations of phylogenetic trees. There we go. Okay, so where has it saved? Tree/Empress. Yeah, sometimes it'll pop up, and if you don't see it, but it says it's saved, you can press the refresh button up there. So Empress, download it. Put in there, and there's our tree. And it doesn't seem like it means a lot right now because we haven't assigned taxonomy yet. But we are able to still calculate this tree even without knowing the taxonomy, because we know we can calculate these phylogenetic differences or distances with a multiple sequence alignment that doesn't require us to have taxonomy assigned. So yeah, I'm trying to like zoom in here. You can like, yeah, you can click on nodes and it'll have names and like it'll tell you how long it is, but they don't mean anything because we don't have taxonomy. So we'll get to that a little bit later, but we also don't need taxonomy to do our diversity metric analysis. So let us run this next cell, Qiime diversity core metrics phylogenetic, and this is going to run pretty much all of the diversity metrics that we are going to use today. And to account for variations in sampling depth, we provide an argument called sampling depth that we've set here to 5,000. We need this to be below the lowest number of sequences or reads in our sample, because this is what we are rarefying to. It's called rarefaction, and it's when you so let's say we have 10,000 reads in a sample, but we're rarefying to 5,000. We're going to randomly draw 5,000 sequences from those 10,000 without replacement. So that we have the same number of reads in each sample, because the sequencing depth or the total number of reads actually really can skew the diversity metrics. For instance, if you had a sample that was sequenced really deeply, it would look really diverse just because you were able to sequence taxa that are really, really rare, even if it's not actually more diverse than another sample. So we should have these. And let's actually go back to the slides and talk a little bit about the statistical tests that we're about to run on diversity. Because we have all of these diversity files, but we now are going to want to test, like, is our diversity different between our case and control groups? And how we can test this is pretty simple for alpha diversity, because you have one alpha diversity metric per sample. So we can treat it as any other kind of sample measurement and use classic univariate tests. A non-parametric test we could use if the distribution does not seem to be normal is the Mann-Whitney U-test, which we'll talk a little bit about later. And a parametric test we might use as a t-test. And it's a little bit trickier for beta diversity because beta diversity is a matrix. So the first thing we might want to do is actually maybe visualize beta diversity. But the issue is that this matrix is the size of it is n however many number of samples we have by number of samples. So here we have 10 by 10. So our distance matrix might be 10 dimensional, and we can't really look at that with our eyes. So what we can do is a dimensionality reduction. And today we're going to be using PCOA, or principal coordinate analysis, which is a linear dimensionality reduction. And that's important because it conserves the ratios of these distances. Other dimensionality reductions might be nonlinear, and so they might distort these. But we want ours to be able to be interpretable. So yeah, it preserves the relationships between the distances in the matrix. And then if we want to do statistical tests for beta diversity, it's a little more complicated than alpha. Not only because it's a matrix, but also because it's very not normal. But we can use Permanova for that, or permutational multivariate analysis of variance. So Permanova is a variation of the ANOVA test that you might be familiar with, but instead of comparing a single variable, it compares the multivariate distances between groups, such as the unifrac distances we calculated. And since beta diversity is often not normally distributed, Permanova uses a permutation approach to generate the p-values. So the first thing Permanova does is calculate an F statistic for the real group labels. In our case, it might healthy versus Parkinson's disease. And then it randomly permutes the labels and recalculates the F statistic many, many times over and over as a sort of control, so that we can then determine the difference between the F statistic if our labels were random versus how they are. And that yeah, to say that in another way, we determine whether the observed difference between groups is greater than would be expected by chance. And the result we get is a p-value, which tells us if this separation between the groups is statistically significant. And how we interpret this a permanova might be in terms of like something like variance explained. So oops, there we go. Let's get back to the notebook and calculate the diversity metrics. Okay, so alpha diversity. We run the diversity plugin and the action alpha group significance. And as a sort of baseline, generally, oops. Okay, so generally healthy individuals have more diverse microbiomes to a certain point. Like a lot, too much diversity might not be a good thing, but generally healthy people do have more diversity. So we've saved this visualization to alphagroups.qzv in a folder called diversity, which we can download. Let's see. And we can look at it. Oops, that's the wrong one. I'm gonna close that. Okay. Oh, sorry about that. That's my notes. That's not the Qiime view. We will find it. Okay. So if you're like me and you closed out of your Qiime view, you might have to use this link to open it again. So let's check that out. Yeah, okay. Oops. We can drag and drop. And Qiime gives you warnings at the top that if it had to omit any metadata columns from this visualization, because as you can see, it does the column or like variable from the metadata, it splits it into however many categories. But if we don't have, if the data isn't categorical, like it's just age, like it could be any integer number, then we don't want like 30 box plots or something. So it'll just omit those. And it also lets you know if you have missing data in certain ones. If there's only like if there's only one group, it's going to also omit that because you kind of want to be comparing between groups. Okay, so if we look at this, we see that it looks like people with Parkinson's disease might have maybe very slightly lower Shannon diversity, although it's not significant. The Kruskal-Wallis test is like it's kind of like the non-parametric version of ANOVA, in that it's going to tell us the distances or the values for all of the groups that we have between other groups, which doesn't make sense now. But when we have more than 2, it might make sense. So our H statistic is 0.5 and our p-value is 0.4. So not even close to being significant or really even trending. Which, you know, it makes sense with a sample size of 10 and a metric as coarse-grained as alpha diversity. You know, we're trying to sum up the microbiome in one number. And that just, there's so much information that's lost. So let's head back to the notebook and we will do the beta diversity calculations. So let's run this. This is our Permanova. We run it using an action called Adonis. And the formula, it can include more variables, but since we only have 10 samples, we're only going to use the one variable, and I think you'll kind of see why. And if yours is still running, we're going to take a minute and actually look at one of the PCOAs just to see what it looks like, although it might not be as exciting as we'd hoped. Okay, so where are the PCOA files? Well, they're in diversity, and they're going to be, let's look at the weighted unifrac emperor.qzv. So unifrac emperor, and I can't tell which one's the QZV, so I can move this bar. So I download this one. Whoa. Okay, so we've got this plot is actually in 3D. So if you click and drag, you can move it around. But usually when you do dimensionality reduction, you're gonna have more than 10 dots because there are some like options we can click here. Like if we want to have different colors for a case control, we can do that. We can rotate it around and look at it different ways. The nice thing about PCOA is that the axes have the variance that is explained by that principal component. So the variance explained by the second principal component is 17.52, and that's the variance explained in the unifrac distances. And this, if it shows clear separation in some way, that can be useful to point out, but what we really want to do is use statistical tests to make sure we're doing robust comparisons. So we generated the Permanova, and let's open that up. So that is at, where did I put it? There we go. Can download that. Oops, I keep clicking on that. Okay. Let's see. Okay, and it just gives us a table. And what this table has is it'll show each variable in our formula. We only had one. And then it shows the residuals, which is everything that's not everything that's left over after we do the association of this variable. So it'll also tell us the degrees freedom and the sum of squares, which is, yeah, what we can use to calculate our R squared, which I think is the most useful thing to look at from a Permanova, because we can sort of interpret the R squared as like variance explained. So here we might say that like Parkinson's disease in this model might be explaining like 9% of the variance in the beta diversity, but our p-value is 0.5 almost, so we can't actually say that. It's not significant, and let's talk maybe a little bit about why. So yeah, go back to the notebook, and I've written out some things so you can read along, but you know, we can't expect much from a sample size of only 10. So even if we could observe trends, we probably wouldn't expect significance. And in addition to that, there are also a lot of confounders because this is an observational study. So they're kind of taking these measurements from people in the wild who are of different ages and live different lives. They have different diets. Diet is a very strong force on the composition of your microbiome. And especially even if we included some of the diet questionnaire things, questions that we had in our metadata, those questions are not very good approximations of what someone's whole diet actually looks like. So because we have all these confounders, and some of these confounders, like diet, might actually have stronger effects on the microbiome than our variable of interest. Parkinson's disease is ultimately a neurodegenerative disorder, and it's related to so many things besides possibly the gut microbiome. So even if the gut microbiome is involved, we wouldn't expect it to be completely causing Parkinson's disease. But if we had a bigger sample size than 10, we might be able to try to deconfound the relationships in our data. And we can't do the full processing pipeline for all of the sequencing files today because of collab and limited computational power. It's the kind of thing you'd want to do on a big server, but luckily I pre-processed the data for you. So, and I've hidden it in the treasure chest. So what we can do is copy the full data set important files into our local directory, like we did if Dada2 didn't work. And I was also holding out on you in terms of the metadata because this study measured a lot of things. So if we read in the complete metadata using pandas in Python, we can see that it's this huge table. It has 39 columns. So that's the number of different variables we have. And that's actually after I manually made it smaller. Since we only printed the head or the top of this table, it only shows 5 rows, but we do now, I did also print the shape of this matrix. So we know that we have 330 samples. And so now we have a little bit more information to work with, and we have the sample size to back it up. So in the original paper, the authors found that some of the things associated with the microbiome composition and diversity were medications like anticholinergics and catechol O methyltransferase inhibitors or COMT inhibitors, and the normal types of covariates like location or like the region of the US, sex, age, and of course, Parkinson's disease. And that some of these signatures are independent of each other, which means that we can deconfound their signals if we do multivariate analysis or stratification of our data. So let's look at how in the full data set, alpha diversity might vary across these variables. We can run this again. We're running the alpha diversity significance testing on the entire data set, Shannon diversities, as we did earlier with the smaller data set. I also want to add everything within that we're doing for the complete study is going to be within a new folder called complete study so that we don't get our analyses mixed up. And if at any point you can't, we've lost you or something can't run, all of the files I've generated are also in the treasure chest, but they're going to be in the folder called complete study outputs. So if you need something, you can also, you want to look at it, but you can't, the collab won't run. You can also download it from here. Okay, I'm going to close it though, because it's secret. Okay, so we've saved this new visualization to diversity/alpha groups. So we can open up the diversity folder within the complete study folder and download that. Wait, yeah, QZV. Okay. And now, well, and this is brings us to the issue on confounding, because it looks like at least with our eyes, we cannot tell any difference between Shannon diversity in people with versus without Parkinson's disease. But maybe there is a signal, but it's being confounded by other variables. So let's look at something else. Seems to be pretty consistent across sex. There's a very strong effect of anticholinergic medications. And yeah, I guess this might be because both of the medications, the anticholinergics and COMT inhibitors, act on neurotransmitter pathways. And these neurotransmitters are obviously in your brain. That's why they're taking these medications for Parkinson's, but your enteric nervous system also uses these neurotransmitters within your gut. So taking these kinds of medications can actually really change the environment of your gut because you're affecting the neurotransmitter signaling there. And I also wanted to note, yeah, you can tell like the p-value of this test for difference is 0.003. But for every different comparison we look at, that's sort of another statistical test that we're running. Even if we didn't like take the p-value from it, we've kind of looked at 3 of them, right? So we'd want to do p-value correction for all 3 statistical tests that we looked at, because otherwise we're kind of cherry picking our p-values. Okay, and yeah, Qiime will also tell you that it has excluded people or samples with missing values in this category, which is nice. Good to know. Okay. Yeah, so we'll go over the p-value correction later, but I did want to point that out while we're in the alpha diversity viewer, just because it is inherently multiple testing. So the next thing we can do, or actually, maybe, yeah, run the Permanova. Okay, and I wanted to mention one more thing. I couldn't include it in this notebook, but one thing we could do with our alpha diversity, if we wanted to deconfound it, is stratify the data. So we could basically split it up into different groups. So how this works is if we wanted to, let's say, exclude the effect of anticholinergic medications, see if that was confounding our relationship between alpha diversity and disease status, then we could use only samples that have that were not taking anticholinergic medications. And to do that in Qiime, we would have to filter the Shannon vector file, which is now a feature in Qiime, but it is not a feature in the version we're using in this notebook, just so you know. So that we just ran the same exact Permanova we ran with our small data set, but for the big one, and it is in complete study diversity Permanova _PD for Parkinson's disease. So let's download that just to look at how increasing our sample size has changed some things. Okay, so we now, the table is set up the same way, but now our squared is a lot less. So we might say Parkinson's disease is explaining about 1.3% of the variance in the unifrac distance matrix. But it is significant. So that's kind of cool. So we needed, I guess, with an effect size this small, we needed this many samples to have to establish significance, which is often the case in microbiome studies. But we think this signal between Parkinson's and the beta diversities might still be confounded by other variables. So now that we have more data, we can actually include these other variables as or in the formula for our Permanova. So instead of just asking the question, how much variance is explained by Parkinson's disease, we would be asking, we'd be kind of splitting up the variance explained. How much is by Parkinson's disease, and how much is by all these other variables, so sex, age, BMI. And we needed more data to do this for reasons that are obvious, but also because with any kind of multivariate analysis, it's not really statistically sound to use to have more variables than you have samples. So like you want to have at least 10 samples for each new variable you add or some other metric. But okay, the first thing we need to do this is actually get rid of missing values, because the Qiime Adonis does not work if you have missing values in any of the variables you want to test. So we're going to do a little bit of data wrangling in Python to figure out where we have missing values. So we print the metadata data frame. And yeah, I've made like a new metadata that only includes the variables we are interested in for this Permanova. And then, okay, which variables have missing values at all? And how many do they have? So Looks like BMI has 11 missing values, and these guys all have 13. This doesn't mean that they're the same 13 people. That just happens to be the case in this case. But so we could have anywhere between 13 samples that we have to remove and the sum of all of these, which, if I can do math, is 40, 50. Okay, so basically a subset of people did not fill out these questions specifically. So let's find out how many samples we need to filter out. Okay, if we remove them, it leaves us with 307 samples, which I think is fine. That means we removed 23 people. But 307 should be good for what we're about to do. So we now have to filter our Qiime table because the table from earlier still has the distances with those samples in it. So there's a specific action in Qiime called filter diversity or filter distance matrix that is going to remake our matrix with the conditions we provide in this argument. We use where, and the syntax for this is actually an SQL. So the is not null is kind of the equivalent to Python syntax of like not equal to, so like exclamation point equal sign. But this will remove all of the, or we're taking out, we're making our new matrix only ones or only samples where these values are not missing, which is some weird double negative logic, but it does work. And it might take a second to run, but once it does, we can run Adonis because we no longer have NAs or null values. And we can use all of these covariates, which should be interesting. So go ahead and run this. And we're calling our file permanova with confounders.qzv. Okay, it might take a second because we put a lot more things in the formula. So it's just thinking about it. And let's go download. Go back here. Wow. Okay. So we can actually see that in terms of variance explained for in the distance matrices, at least, our variance explained by Parkinson's disease didn't change a lot. So actually, we're seeing distinct signals for all of these variables, which is cool. And some of them are significant, like location, it looks like, which could have to do with a lot of things, like the people were in either Seattle, Atlanta, or maybe Albany, New York. Lifestyle and diet can vary a lot between those places. And it also might have to do with what hospital it was. So yes, now we have these. So that is how we might do some sort of deconfounding. And we are just about done, but yeah, or with this part, we'll take a little break. But if you want to during the break, you can check out the full PCOA in the visualizer. And it's like the last one, but it has 300 dots instead of 10. So it might be more interesting to look at. You can mess around with the coloring, but yeah, we're gonna take a break and see you back here in about 20. Hope you have had a nice break. And if you'll join me back in maybe we should start in the notebook, because we're gonna start running this cell and then we'll talk about some things. Okay. So, well, I'll give you a second to, sit back down. But if you recall, we started with raw sequencing reads. We imported them into Qiime 2. We denoised them with Dada 2 and built a phylogenetic tree from the ASVs. Then we did diversity metrics. And on those, we calculated or did statistical testing. So we still have our representative sequences or ASVs, but now we want to go from sequence to taxonomic assignment. This way, we can identify specific bacteria that might be enriched or depleted across interesting variables. So if you will join me in the notebook, you will see this new section called taxonomic classification. We are going to run this first cell. And while it runs, let's talk about what's happening in the slides. Okay, so what organisms are in our sample? Up until now, we were just working with sequences. Even though they were cleaned and denoised, we don't know what actual organisms they are. So the question is, how would we go from sequence to an organism's name? And some background on taxonomic rank is that taxonomy is a hierarchical structure that gets more and more specific as you go down. So high level, or the highest level, is domain, which here we might be talking about, but for this fox, it's a eukaryote. And then we go down to kingdom, all the way down maybe to species. So an important note here is that the lowest taxonomic rank that we can confidently assign our 16S reads is the genus level. Here it's the kind of purpley red one, because basically there isn't enough diversity in the 16S gene to distinguish between species. But there is between genera. And with what we're talking about, the variation in the gene, there is still a lot of divergence, even within a genre or species. And this can cause issues if we're trying to map the sequences that we have in our sample back to a reference database, which is only going to have reference genomes. We can't have reference genomes for everything that exists ever. So we're going to have different sequences that don't exactly match the genre even that they might be assigned to. So we might think that directly aligning our sequences makes the most sense, but this might not actually work very well because the sequences we have might not match this reference, even if they should be in the same genre. So we need a more generalizable approach. Instead of looking for exact sequence matches, maybe instead we could look at if the sequence is generally conserved. So, oops. What we use to do that is called a multinomial naive Bayes. But this all looks very complicated, but it's actually just a little bit of clever math, and we're gonna walk through it. Okay, so let's say we start with a query sequence. So that is here. Our sequence is ACG, CGC. We can divide this query into subsequences of the same length. Here we're doing 3 mers, so a 6 base pair sequence can be split into 4 3 mers. And if we subdivide our query into all possible k-mers, which our actual query is going to be a lot longer than probably just 6, yeah. But that builds us a profile, basically, of how many times the 3-mer ACG occurred. And that profile, it turns out, is pretty taxon specific. So how we actually go from this profile to mapping is with Bayes' theorem, which is here. So Bayes' theorem tells us that the probability of finding a query sequence, oh wait, okay. So the probability of this, of it being a taxon given a specific query sequence is equal to the probability of finding the taxon at all, multiplied by the probability of it being the query given we already know that it is this taxa, divided by the probability of finding the query sequence. And if we assume that the probability that we find the query sequence at all is about the same across all bacterial taxonomies, then we can kind of simplify this and say, okay, the probability of it being taxon 1 given our query sequence is approximately equal to the probability of it being taxon 1, which is a prior that we should already know before we sequence. So like how prevalent is this taxon usually, multiplied by all of the probabilities of the individual k-mers. And that gives us the probability of it being taxon 1 given our query sequence. So we then can select which taxon is the most likely based on this number. So here we would assign this sequence to taxon 1. And this provides us a more generalizable approach than mapping directly to the reference genome, but it also is a lot faster, and it uses machine learning, and it actually has a pretty good accuracy. So let's try it out. That's the cell we ran earlier. Head back to your notebook and let's see what we just ran. So we ran the pipeline or plugin feature classifier and the action classify sklearn, which is actually, it's using a package from Python called scikit-learn that anyone who does machine learning might be familiar with. So we give it a pre-trained classifier, which we already had in our files. These classifiers are pre-trained to assign bacterial taxonomy. because training them is really computationally expensive. We don't want to have to be training them every time. So the one we chose is specific to the V4 region for 16S data. And then we get our classification out. It's a Qiime artifact called taxa. And if we want to visualize this, we can make a bar plot out of it. Qiime has a pretty cool bar plot for taxonomic assignments where you can select what taxonomy level you want to see. And if we download this from, let's close that, from our materials folder, it'll be directly in that called taxabarplot.qzv. So we download and we can open it up. Okay, so this is a little scrunched up here on the side. We can make it a little wider by dragging out the bar width. And we are currently at the highest taxonomic level. So this plot is telling us that all of our samples are entirely bacteria, which is expected because we isolated bacterial DNA. But if we want to look at the composition on the genus level, that would be level 6. Now that is a little more interesting. So all of the different genera are labeled here, and you'll notice that some of them are unassigned. So this one is an unidentified genus within the family of Enterobacteriaceae. So yeah, not all of the taxonomies could be assigned because there did not seem to be a genus that fit within our reference database. So if you, I guess this key does reuse colors. So you can tell what a taxon something is by scrolling over it, and it'll appear here at the top just to make sure. So this is the one we just pointed out. And then we have a Bacteroides. So this makes a lot of sense that we have sort of a split between people who are Bacteroides or dominated by Bacteroides versus dominated by Proteobacteria. This is something we commonly see in the microbiome. But if we wanted to look at this data and organize it by some kind of variable, we can do the sort samples by and choose one of our metadata variables, like Parkinson's disease. So now we're looking at this. The healthy controls are on the left, and Parkinson's patients are on the right. And just looking with our eyes, we can't really, I mean, at least I can't pick out a sort of trend here. It's, yeah, we would have to maybe do some kind of statistical testing to identify trends, especially if they are of small effect sizes like we've previously seen. You know, last year we did C. difficile infection, and we saw a very, very clear trend between people who were infected versus not infected. But this signature is a lot more subtle than that. So just plotting our taxonomic abundances doesn't tell us very much. So let's go back to the notebook, and we will, we're going to collapse the table onto the genus level, which actually I'm just going to show you. You can download the genus level table as a CSV from here on the page, actually, or you can use a command in Qiime to do it, which is what we're going to do. So we say taxa collapse, and we specify that we want the genus level, which is level 6. And then we're going to put it in a file called genus.qza. So let's run that. It should not take too long. And what we, so now we have this Qiime artifact that contains our genus abundance information, but we want to turn that artifact maybe into a table that we can work with in Python or something. So we can actually export data from Qiime back to a table like a TSV or Excel using Qiime tools export. And this automatically exports our table to a .biome file, which is a specific type of, or file type for taxonomic abundance tables. It's actually, it's a pretty strange file type, so you can't just maybe open it as just one table. It actually contains the taxonomy information in a sort of separate object. So what we are doing in the second line is converting the biome file into a TSV, which that will give us just a single table. And then we can import that TSV into our notebook using pandas read. So let's read it in and check it out. Okay. So we can see our row names or index is called OTU ID, and it has all of the taxonomic levels and the genus level at the end. That's the lowest one we went to. And the columns are our different sample IDs. So this might be easier to interpret by plotting. An example of something we could do is maybe use some kind of visualization tool in Python like Seaborn. So here's an example of that. We can import NumPy and Seaborn, and NumPy is going to let us do some different math to our abundances to make this plot look nice. And we're going to talk a little bit about the transformations that we do in a minute. But we can see here, we've normalized the abundance to make it centered around 0. And we have all our samples here. So this is just kind of like a different way of viewing the bar plot that we looked at just a minute ago. And it still doesn't mean a whole lot to me, at least. But, you know, it might be interpretable if we work with it a little bit. But to work with it, we need to transform it in some ways. Because the first issue is that if we scroll back up to our table, these are not relative abundances, but counts of how many times this taxon was observed. And that number is not comparable between samples because we had a different total number of counts between samples. And so let's go back to the presentation to talk a little bit about compositional data and some of the challenges it brings. Okay. Okay. So we want to compare abundance between groups of samples, but our abundance is compositional. It's relative, maybe. The first thing that's important to understand is that sequencing is an indirect measurement of what we actually want to study. So we want to know the information about the whole ecosystem, but sequencing takes a random sample of organisms from our sample. And because of this, we don't know the absolute abundance or even the concentration of any bacteria in a sample. We only know their proportions relative to one another. So in this plot, which is from a 2017 study, they actually used a sort of quantitative method to determine the total concentration in a sample of bacterial cells, which is also kind of, those methods can also be biased, but it does show us that, we actually have one sample that has a pretty high concentration, and another, this might range from, samples being like 6 times as concentrated. So even though, like this, what was I thinking about? Yeah, so with what we actually are looking at when we do relative abundance from our sequencing data, we don't have this information. We only have what's in this red box. So it looks like our last sample here has a ton of this purple bacteria, but in reality, it actually has way less than the first sample. And there's not really a way for us to know, other than trying to measure concentrations, how much total was there. So what we're actually analyzing is not how much was there in the first place, but how abundant is a bacteria in relationship to all of the others. Assuming that these proportions are comparable and not confounded by some kind of taxon-specific sequencing bias, which it turns out might be a problem. So just like we chose earlier to use genus level annotations, let's continue on with that. We could analyze differential abundance at any taxonomic level, but it kind of, what you choose might depend on what questions you want to ask and what type of data you actually have. So next question, what statistical tests can we use? Because, and this is kind of foreshadowing that we might not be able to use a simple t-test on the relative abundance data. And to know what test we can use, we have to look at how the data is distributed. And ideally, we would also look at the variance, but so microbiome relative abundance data, and the relative abundance that I'm talking about here is the count of one genus divided by the count counts of all genera in a sample. So the value of relative abundance is going to be between 0 and 1. And the issue with this specific data structure is that, well, 1, it's compositional. Every variable depends on every other variable because they all have to add up to 1. It's not normally distributed, as we can see in this plot. And even if it kind of looks like it might need a log transform, but if we look at it on a log scale, we see separation of the 0 values and the samples in which it was measured. And this is because our data is 0 inflated. So it contains both true zeros and zeros that are created by sampling. So a person might have had a certain microbe, but we didn't sequence deep enough to find it. And there's no way to know which zeros are true or not. It's also, I didn't graph it here, but it's more variable relative abundances than we would expect in a Poisson model, which means that it's over dispersed. And this is partially because of clustering effects that might actually be derived from true ecological relationships. So for example, the chances that you would randomly draw one microbe might be higher if you had already drawn another microbe that the first one is like feeding off of a substrate that the second one produces. And this is because of cross-feeding. So it'd be more likely that a person either has both or they have none. They're not randomly distributed. So you have more or higher variance. And all of these things together, including this, we still have the issue of not normal distribution. These are going to violate the assumptions, at least of parametric statistical tests. So what are we going to do about it? Well, we have a few options at this point. Our first option is to use relative abundance data, stick with that data and just run non-parametric rank-based tests. And this is usually a good first pass for differential abundance analysis, but it is going to be underpowered because we're not making assumptions about the distribution of the data. The second option is that we could transform the data in some way. But as you saw in the last slide, if we still have the issue of the 0 inflation. So when we transform the data, we're either going to have to impute the zeros. So we could kind of make up the values, or we could discard them entirely, and then we just lose that information. But if we can get our distribution to look normal, then we can run parametric tests, which are going to have higher power, but we're going to be asking a different question because we got rid of the zeros. The third option is that we might use a more complex modeling package, all of which have their own caveats and assumptions. And there are many out there, and some of them do different transforms. Many use a pseudo count to impute zeros, and it's very specific to whatever package you're using. So today, I just am going to show you how to work with the data with the first two methods, to look at it manually, to really give you a good understanding of what's going on, or at least the basics of what might be going on under the hood with these more complex methods, because it's important to understand what you're actually doing, especially when you're making assumptions in statistics. So for the first option, we're not going to transform the data. We're just going to use a non-parametric test. And specifically, the Wilcoxon rank sum test or the Mann-Whitney U test. They're the same thing. And this is kind of the most basic type of differential abundance analysis we can do. And we'll be looking sort of, or we're comparing we're trying to decide if the two distributions come from the same population. Except we're not looking at it on the scale of the actual relative abundance. We're ranking, we're using ranks instead. So the ranks will range from 1 to however many taxa were in a sample. And by going from relative abundance to rank, we kind of normalize this curve, but we also do lose the data that is the relative abundance, like actual ratio. But it is a good first pass. So let's actually go to the notebook and try that out. Okay, so we're gonna look at the abundances in the complete data set, just so you can really see the distributions, because a histogram with only 10 samples does not really show you the distribution very well. So we're gonna import some more packages that we're gonna be using. And before we do anything, we need to make our count data into relative abundance data. So we divide, oh yeah, so this is just us reading the table for the complete study instead of the smaller one. But what we do is we divide these by the sum of each column, because each column is a sample. So we're normalizing it to the sequencing depth of that sample. Okay, so this will give us a new table, and the values should look a lot smaller. And you'll notice, just like how in the plots we had a lot of zeros, the plot I showed you in the slides actually was from this data. You can just see how sparse some of these columns are. So pre-processing steps we might do are definitely get rid of samples that had no taxa assigned. This could be if you had if somehow a blank got through the rest of your processing. But with all the analysis we did in Qiime, we know that none of our samples should be empty. So that's good. Then we might want to maybe remove taxa that are not present in most people. So let's say we want to set a cutoff, like if a taxa is only present in 20% of our samples or less, then we're just not going to look at it. We're going to take it out of this data frame. And that's what this cell is going to do. And I also flipped the data frame so that our samples are rows and our columns are the different genre. So because this matches the format that our metadata is in, if you recall. So now let's save the column names of the genre in a list so that we know which columns were bacteria, because we're about to merge this table with our metadata table, which has a lot more columns. So we don't want to lose track of all of them. Okay, that has run. And then just to show you one taxon in this table, we can plot the relative abundance and split it by the disease status. So this is like the plot I showed you on the last slides, but for Faecalibacterium. So you might look at this and think, Well, it looks like people with Parkinson's seem to more commonly have no Faecalibacterium. But how do we know if this is significant? Then we can do our nonparametric test if we don't want to transform the data. So if we want to know if it is truly differentially abundant, then we split, if we're doing a Mann-Whitney U-test, we need to split the variable of interest into two different lists because that's the kind of input that this test takes. And these column names are so long. But we are just saying the first one is the Faecalibacterium relative abundances for people with Parkinson's, and the second is Faecalibacterium for samples without. And then we're gonna gather up our statistic and p-values and print it. So we actually see that even using a sort of underpowered nonparametric test, our p-value is significant. So it would seem that Faecalibacterium mean relative abundance is significantly different between Parkinson's disease and healthy controls if we were using a cutoff of 0.05. But if we wanted to do some different types of analyses, then we're going to have to do more transformations. So let's head back to the slides to talk about it. Okay. So one normalization method that accounts for the compositionality of the data is the center log ratio transform, in which the counts are not only log transformed, but also divided by the geometric mean of the sample, which the geometric mean is different from the arithmetic mean because it is less sensitive to outliers and gives more weight to smaller values. And you'll see here, it's actually the nth root of all of the accounts or abundances multiplied together. So we take the log of one count over the geometric mean of the sample, and that will give us all of our CLR transforms for one. Oh yeah, so so geometric mean of the sample. So this will give us our CLR transforms for that sample. So we need to do this transform to every sample. And when we do that, it's still not going to look normal because we have those zeros. They're not going to be one value anymore because of how we did this normalization. It's going to look kind of bimodal. So we either need to we could impute these like on a maybe a random distribution before we do the CLR transform of values that are small. So we're just kind of adding noise, or we could just get rid of them. And that's what we're going to do today. So we'll just remove the samples that had 0 reads for this genus. And then we're left with only this second peak, which is here. And it looks kind of normal-ish. And then with that, we can do some parametric tests. But after we test, we still need to correct our p-values. And like I mentioned before with the Kruskal-Wallis test for alpha diversity, even the fact that we just looked at 4 different tests means that we did multiple testing. If we are going to test every single genre for relative abundances, then that means we'll have done like 100 tests or more. And when we do multiple tests, if they all have an error rate of 5%, then if we did 100 tests, we would expect 5 false positives. So that's this in this figure. If there's no true relationships, we're still going to have these false positives. But when there are true relationships in our data, the false positives are combined with the true positives are going to really inflate that part of the p-value distribution. So the goal of p-value correction is to set a different cutoff for our p-values than 0.05 to try and cut out some of this red. So there are different methods that kind of weigh the balance between how many false positives are allowed and how many false negatives we are okay with. When we're doing kind of initial biomarker discovery or something, we are okay to allow some false positives because we want to capture all of the true positives. So we might do something like the Benjamini-Hochberg transform, which I will show you. And let's go back to the notebook and do it ourselves. Okay, so before we can do a CLR transform, we have to add a pseudo count, which is just we're adding some really small value to every single value in our data frame because we can't take log 0. It does not exist mathematically. So, but before we do that, let's actually keep track of which values are non-zero so that we remember, and then we can cut out the zeros. So we do this. We saved which ones were 0 for later, and then we added a pseudo count. So now we should see no zeros, and the lowest it'll go is e to the -7. So now that we have pseudo counts, we can do the CLR transform, which we can do using the SciPy stats package. But they have a function called gmean, which is geometric mean. We compute this. So the CLR, we're doing along each row. Every row is being, the values being divided by the geometric mean and then the log taken. So our output data frame is going to look, the values are going to be somewhere between, -10 and +10, but usually, because we've transformed it to a different scale. And to make sure, actually, to make sure that the CLR went correctly, oops, I usually like to, at the end, I leave the gmean column on for a second to make sure that when I took the log of gmean divided by gmean, that equals 0. That means I did it right. So we're safe. That means we can remove our gmean column. And now let's look at what that transformation did to the Faecalibacterium we plotted earlier. Okay, so now we have our CLR transform data with our bimodal distribution. So like we kind of talked about, the most conservative option that we can do is remove the zeros, because at the end of the day, we don't know if they're true zeros or not. So there's always going to be some kind of uncertainty around that. But that means that the question that we're asking then might become, in people that have measurable amounts of Faecalibacterium, is there a difference between groups? And that's a different question than the one involving zeros. So let's drop those samples by making them null. And then we can plot again and see that the distribution looks a lot more normal than what we started with. It's still a little skewed, but it's probably safe to do some parametric tests. So this means we can merge our CLR transformed data back to the metadata because we need to match our samples to their metadata information. So I'm making a new data frame called merged CLR. And you can see we've added on all this metadata, and then at the end is where all of the CLR values are. And so any value that was 0 is going to now be null, or NA or NaN, which is not a number. So the code to perform a t-test is very similar to that of the Mann-Whitney U-test, but as we did with the Permanova in Qiime, we need to remove those NAs because the test can't just ignore them. So what we're doing is this kind of the same code, but at the end we're saying drop NA for the column. So the number of samples that we're testing total is not going to be equal to 330. It's going to be less because some of our samples have NAs. Okay, so for this one, we actually still have a significant p-value, which, you know, if we were truly doing analysis, we would have to correct all of these p-values once we're done. But this, yeah, so this statistic from a two-sided t-test tells us that in people with measurable amounts of Faecalibacterium, people with PD have significantly lower Faecalibacterium relative abundance than people without PD. So the question is a little bit different, and it doesn't include anything about the people who have none, which is also different between the two groups. So to correct for multiple testing, we are first going to do all of the tests and kind of put them all into one, put the results in one data frame to make it easier to correct to correct the p-values, because the correction method we're using, Benjamini-Hochberg, uses, it corrects the p-value based on all of the other p-values. So we need to be using all of them anyways. Okay, so this code is going to run all of our tests for each genus. And then I convert the results into a data frame. And let's take a look. So every genus is in the column genus, and then we have a t-statistic, a p-value, and I included also the number of samples that were in each category, because as we mentioned with the NAs, it's going to be different for everything. And if we had really like unbalanced samples, that would kind of affect how we might be able to interpret this data. But it looks like most of them seem not necessarily balanced, but like we have enough samples in each to really get a good distribution. Like if one had only one sample, that wouldn't be a very good test to be running. Okay, but what does this distribution actually look like when we compare between two groups? Let's plot it. Or actually, yeah, no, sorry. What does our p-value distribution look like compared to the one we looked at on the slide? We can see it has a leftward skew, and that is good. That makes sense because we expect false positives and true positives. It's a little a little wavy here, but it should be okay. And now that we have the list of p-values in a data frame, we can just easily do this correction. So we're going to use a function from stats models, multiple testing, and the method is just FDR, which is false discovery rate, and BH is for Benjamini-Hochberg. So we gather our Q values, and then we're going to just append them in a new column to that data frame. And then I'm sorting the data frame by Q value so you can see the significant hits. So you'll see at least the first 5 hits here are significant after p-value correction. And since the genus column is cut off, we don't know what genus they actually are yet. But let's make the names a little easier to plot, and maybe we can visualize this data. Okay. Okay, so here I have changed the genus label that I'm going to use to plot. So I've taken only the slice of the string in the genus column that contains the family and the genus, because as we mentioned before, some of the genera are not annotated. So I didn't want this one to just have like a blank as for what it was. But to visualize this information, this is a pretty, I was doing some like extra fancy things with this plot. You don't need to do all this, but one way we might visualize it is plotting the t-statistic for each hit, and maybe we could color it by Q value. Maybe that would be informative, and to have the labels. So maybe this is easier to look at than the table. We have darker Q values are more significant, lighter are getting closer to our cutoff that we used, which was 0.1. And we have the t-statistics. So all of these bacteria are less abundant typically in people with Parkinson's. And if you are a microbiologist, you might notice, oh, some of these bacteria are thought to be doing good things in the gut microbiome. So that could actually be an issue. And that kind of ties back into the original analysis that this paper did and the meta-analysis that they found potential like decreases in bacteria that are generally promote anti-inflammation. So you might have a more inflammatory type of microbiome if you have Parkinson's disease, which could be contributing to, definitely contributing to GI symptoms. And it could possibly be contributing to things in the brain as well. We don't really know. So this is how you might do a really basic differential abundance analysis. And I'm going to head back to the slides to show you like one more slide, because the limitation to differential abundance analysis is that the assumption we made earlier that the taxonomic proportions of an ecosystem are conserved once you've done your sequencing, that might not be true. So in this paper from 2019, an analysis of defined bacterial communities suggests that bacterial taxa are captured by sequencing at different efficiencies, and that this bias can distort differences in observed relative abundance between samples or groups of samples. And this figure, okay, sorry. This figure is from this paper, which is from the Willis lab at University of Washington. And here they demonstrate that if you have known actual relative abundances, so that's on the left, and you might have taxa that are sequenced at different efficiencies. So the efficiencies are 1, 18, and 6. So we have a low, high, medium efficiency. So you can see here in sample 1 that we have equal parts of all taxa. And we definitely have more of that high efficiency taxon than the other sample. So what we see is that because that high efficiency taxon is pretty abundant, it gets super overinflated in our observed relative abundance. And that causes the ratios of the less efficient taxa to shrink. And kind of the same sort of thing is happening here, except that we have a little bit less of the really high efficiency taxon 2. So it takes up less of our observed, which really lets taxon 3, which is the medium efficiency, actually kind of grow or appear to take up more space in our observed relative abundance. So even though we know that taxon 3 is more relatively abundant in sample 1 than sample 2, if we only looked at the observed relative abundances from sequencing data, we might think that taxon 3 is more abundant in sample 2. So this kind of bias is still an unsolved issue in sequencing data because we don't know the sequencing efficiencies of different taxa. It comes from a lot of different steps in the pipeline, like how many of the 16S copy number do you have? Different taxa might have between 5 and 15 copies of the 16S gene, and so that can distort things, as well as differences during, like DNA extraction even. But I did want to shout out a new method that tries to account for this bias that's in review. It's called RAD-EMU, and it's developed by David Clausen and Amy Willis here at the University of Washington. And it's available as an R package on GitHub, which is why I'm not demonstrating it for you today, because it is in R. But I do use it in my own analysis, and it's available publicly on GitHub. Oh yeah, so we already did our normalization and testing, but the conclusion from all of it and plotting along the way would be to always look at your process data, even if you're running a more complex program like RAD-EMU or something, because not all models for differential abundance testing are going to be checking to make sure the assumptions they're making are true. So that's where you might want to be checking some things manually. Yeah. So there's one activity in the notebook if you want to try. It is to add the taxonomic assignments to our tree and answer some interesting questions. You have to fill in the blanks with the files that we're going to use, and just a warning, use the files for the small data set, not the big one. Otherwise, it might not run. I don't know. But yeah, we're going to break now for lunch, and thank you so much to everyone who helped put this together, and thank you guys for showing up. Oh, yeah. Okay. So one more thing is that after our lunch break, we are going to have a talk from another member of the Gibbons lab, Jacob Cavon. He's going to talk about microbes that might be differentially abundant and the metabolites they are making in perhaps relation to sleep.
My name is Jacob. I'm a second year PhD student in the Gibbons lab. And today I'm going to be talking about my research on using wearables data to look at the relationship between sleep and the gut microbiome. Thanks for joining us today, and thank you to Alyssa for the great lesson, the introductory lessons on data analysis. Hopefully my presentation will be of interest, and maybe you'll recognize some of the data analysis techniques that you learned this morning. Also, if you have any questions, be sure to write them into the Slack, and then we can get to a Q&A session after my talk. Oh, on Zoom, sorry about that. Yeah, please write them into the Zoom Q&A. Thank you. Okay, so before we get into the weeds, let's just quick refresher. How does the gut microbiome talk to the brain? This morning, if you remember, Alyssa showed a really great cartoon of all the different ways that the gut microbiome interfaces with the central nervous system. And the way that it does this is through signaling molecules that it produces. So if you remember from this morning, some microbial metabolites can diffuse across the gut lumen and directly enter systemic circulation through the hepatoportal vein. Actually, one study from our lab found that the gut microbiome significantly contributes to the cross-sectional variation in 44% of measured blood metabolites independent of host genetics. Another way that the gut microbiome can communicate with the nervous system is via metabolites that it produces that interact with vagal afferents. And so vagal afferents are, afferents here means that you have the vagus nerve that connects the enteric nervous system to the central nervous system, and the afferents are nerves that connect signaling from the gut to the brain. So some of these microbes actually produce human neurotransmitters like GABA that interface with the vagal afferents, and those signals are relayed to the brain. And then third, some microbial metabolites can interact with peripheral immune cells, and they can influence the brain through immune axes. Importantly, many microbial metabolites actually remain functionally uncharacterized, so we don't actually know what they do in terms of affecting host physiology, and there are many that are yet to be discovered. Okay, so you might ask, there's some research on how the gut microbiome interfaces with the central nervous system, but what about how does the gut microbiome influence sleep? Well, this is an emerging research area, and there are a handful of pre and probiotic randomized controlled trials that show some promising results in supporting sleep. So I want to walk you through one recent study by Fei et al. From 2023. This was a 12-week probiotic intervention where participants took either a lactic acid bacterial probiotic or a placebo pill once daily. And you can see here the way that they are, their outcome variable is the PSQI. This is the Pittsburgh Sleep Quality Index. This is a self-assessed sleep questionnaire. It's been demonstrated in a number of different cases to be valid and reliable in assessing sleep. And so you can see here, if we look at column D, this is the difference between the probiotic and the control groups at week 12. There was a significant improvement in the PSQI total score in the probiotic group relative to the control. And so here you have the total score where a bigger score means worse sleep and a smaller score means better sleep. So you can see for instance, in the probiotic group relative to their own baseline, their sleep significantly improved. In addition, if you can compare the placebo group to the probiotic group, you can see that the probiotic sleep is much better compared to the placebo group at week 12. And then down here, we just have, this is a breakdown of the components of the PSQI. So you can see things like, okay, sleep quality improved, sleep duration improved, time to fall asleep, etc. This study also looked at gut microbiome 16S sequencing at week 12, so at the end of the intervention. And they compared the placebo group to the probiotics group. And here they used a Wilcoxon rank test, which maybe you remember, Alyssa went over this. This is a non-parametric test to compare relative abundances. And you can see that the probiotics group are enriched in several different genera, including Blautia, Coprococcus, Ruminococcus, and then Prevotalaceae, which is a family, and Lachnospiraceae. And then they also looked at blood serum, and they found that the probiotic relative to the placebo had higher blood plasma levels of BDNF, which is an anti-inflammatory molecule. This stands for brain-derived neurotrophic factor. So there's some interesting things going on here. Patients took the probiotic, their self-assessed sleep was improved, and then at the end of the trial, certain microbes were enriched in the probiotic group relative to the control, along with an anti-inflammatory molecule. So perhaps there's certain gut microbes that are affecting immune signaling, perhaps through an immune axis that affects sleep. But we really don't know what signaling molecules is the gut microbiome producing to influence sleep, and what host machinery do these molecules actually affect and how. So how are we going to start getting at this question? We can start by leveraging large multi-omic data sets. So here I'm going to run you through the approach on how we get at this question of mechanistically, how does the gut microbiome affect sleep? We could start off with this sort of reverse translation approach where we look at these large human cohort data sets, and we use statistical analyses to mine associations with different variables with our variable of interest. So maybe we're interested in sleep, and we want to find associations between certain microbes or certain metabolites and sleep. We can then take those associations, make our hypotheses, and then we can test these hypotheses in with experiments, either in cell culture or in animal models. So maybe we think that bug A is able to metabolize some compound here, and then that metabolite can go on to affect sleep. We can maybe mix this bug with this metabolite and see if the metabolite changes. And then maybe we feed this bug to mice and see if mice sleep longer or something like that. And then once we have a clear picture of the mechanisms that are underpinning these associations that we found in the human data sets, we can use that knowledge to design interventions to treat our variable of interest, and then test our interventions in human trials. So today, I'm just going to be focusing on this first part of the approach, which is mining large human cohort data sets to find associations between variables that we can use to generate hypotheses to test. So in my research, I've been leveraging Airavail. This was a health coaching program, and we have this large, deeply phenotyped human cohort data set now. There were around 1,500 participants, and lots of different data was collected from these people, including 16S sequencing data, blood metabolomics, things like BMI, sex, age. In addition, participants were required to wear Fitbits. So we have wearable data on their sleep and activity habits. And then there's also extensive questionnaires ranging anywhere from like mental health questionnaires to diet. So before we get any further, for my analysis, I just mentioned that the participants were going to use Fitbits or used Fitbits. So we have the sleep data, and that's the data that I'm going to be looking at for my analyses. So you might have some questions about whether Fitbit is a reliable, can reliably estimate sleep. Here's a recent study from 2021, and I'm just going to walk you through this. So these plots are called Bland-Altman charts, and they are used to look at the agreement between two kinds of measurement tools. So for instance, in this first chart here, we are comparing ActiWatch to PSG, which is polysomnograph. If you're not familiar, polysomnograph is the clinical and research gold standard for measuring different sleep parameters. So this is, if you yourself, or maybe you've had a family member who's been referred to a sleep doctor, maybe you had an overnight sleep study done. That's where they use polysomnograph. So they attach electrodes to your head. They also measure other things like your heart rate, your breathing rate, your muscle tone while you sleep. And so polysomnograph can estimate total sleep time, or it can measure total sleep time, sleep efficiency, time to sleep onset, and wake after sleep onset. But in addition to polysomnography, clinicians and researchers also use actigraphy, which is a watch that you wear that estimates your sleep based on movement. And in the clinic and research, this ActiWatch is used to do that. So if you go to the doctor and they need to do a sleep assessment, they will likely give you an ActiWatch to wear and then do a PSG if that's required. Okay, so let's see how the ActiWatch compares to the PSG. Remember, these are both the clinical and research gold standards. And then we'll look at how the Fitbit compares to the PSG. So this dashed line here, this is called the bias. And you can see that, so each of these dots is 1 night of sleep measurement. And then here we have the difference between the ActiWatch's sleep measurement and the PSG's measurement. You can see that the ActiWatch on average estimates total sleep time about 30 minutes longer than PSG. And then down here, we've taken the average between the ActiWatch and the polysomnograph measurement for each night. And this kind of just gives us an idea of the range of values of total sleep time. So you can see that if total sleep time is less, the ActiWatch actually tends to overestimate total sleep time compared to PSG. Now, if you move over here to Fitbit, you can see that the bias is a lot closer to 0. So there is, so that means that whatever the Fitbit is recording for total sleep time is not, it's pretty much the same as what PSG is measuring. And then you can also see that as we decrease total sleep time, there's less variability here. So the Fitbit is better than the ActiWatch in terms of measuring total sleep time, even when total sleep time is less. We can take a look at these other plots too, like sleep efficiency, and we generally see that there's less bias in the Fitbit data compared to PSG than the ActiWatch compared to PSG. So that's great news. So Fitbit is a reliable measurement tool that is in close agreement with PSG. Okay, so let's go back to our Airveil data set. So I'm going to focus in on these columns from our data set. We have 921 different blood plasma metabolites, 74 different gut microbes, and 6 different Fitbit sleep measurements. These are the columns I'm left with after doing some data wrangling. Earlier today, Alyssa talked about data transformations like CLR transforms for the gut microbes, and then dropping columns that are sparse. So I've dropped columns that are missing more than 25% of the data. We also do some log transforms for the blood plasma metabolites and the Fitbit sleep measures so that they look more normal. And then we can take all of these columns and we can apply regression and causal mediation analyses to find our associations. And this narrows down our what I'm calling hits to around 20 hits, where we have 20 different relationships, statistically significant relationships that we found. And then we can take those relationships, generate hypotheses, and then collaborate with other labs to test these hits in cell culture and animal models. Okay, so let's work on the first step of our analysis, and that is to build sets of regression models for each combination of features. So first, we're going to look at, we're going to build regression models for gut microbes and sleep. So we're trying to regress sleep on gut microbes and then control for different covariates. We then save the statistically significant hits, and then we do the same thing for blood plasma metabolites and sleep measurements. We take the hits from 1 and 2, and then we run those regressions against each other where we look at whether a gut microbe is associated with metabolite after controlling for these covariates. So this is kind of what it looks like to make the sets of regressions and then the formula for each regression model. So we have several sleep columns or variables that I mentioned earlier. I think there's 6. So for instance, there's like restless duration, minutes of sleep. And then for each of those, we're going to run a for loop through every single gut microbe that we have. So there's 74 different gut microbe columns. So we have like actinomyces, Clostridium, Adlercrucia. And then we do the same thing for the next sleep variable, etc. Then each of these regression models, we add the following covariates: sex, age, BMI, ancestry, and then 10 different Fitbit activity features. We're including these covariates to control for confounding because we know that activity for sure affects sleep, and so does age. Yeah, and then sex and BMI. So after we do this, then we have all of these different regression models where the formulas look something like this, where we're regressing a sleep variable on one microbe, and then this set, we're controlling for this set of covariates. And then we can fit the models to our data, and we get our coefficient estimates. And then Alyssa was mentioning this before on how we can control our false positives. So we can set our false discovery rate to 5%, and then we save those coefficients. So whatever microbe, we save those coefficients that pass the 5% FDR. Okay, so once we've ran our regressions, the important thing about the regression models is that we can use them to control for confounders. And we see that there's like, for instance, the microbe is significantly associated with sleep, even in the after controlling for the confounders. So after we do that, we can make a summary plot like this. These boxes are Pearson correlations between the two variables. So this is just showing the strength of the relationship between the two variables, either positive or negative. So the direction as well. And then if there's a box, that means it was a significant hit from a regression model. So we see several significantly associated bugs, including Ruminococcus, Roseburia, Coprococcus, Blautia, Lachnospiraceae. If you remember, this is consistent with that probiotic study we talked about earlier, and it seems like Lachnospiraceae members are generally associated with sleep outcomes across several pro and prebiotic studies. We also have these guys, which are also implicated in those studies, as well as Adler-Crutzia, which there's not a lot of literature on this guy, but the existing literature is kind of interesting because it suggests that it definitely has anti-inflammatory properties and may have some neuroprotective effects. Okay, so then we run our set 2 of regressions, and we can make a summary plot of these guys. So we have several, this 2-piperidinone. If we do a quick just literature search with some keywords of these metabolites in sleep or brain, this is a good sort of like gut check that maybe we're on the right track here. So here it's like, okay, S-adenosyl L-homocysteine increases REM sleep. There's some study here looking at it in Alzheimer's. Piperidine is an upstream molecule of 2-piperidinone. We can see, okay, N-acetyl aspartate, which is here. There's some relationship here between Ruminococcus and then the brain N-acetyl aspartate in pigs, etc. Okay, so then finally, after we have our metabolite sleep and microbe sleep hits, then we run these against each other. And we see if we can regress metabolites on microbes. So let's look at all the associations here. So cool. We have like, okay, some associations between N-acetyl aspartate with these guys down here, Clostridium. And then we can take, so that was the middle plot, was what we were just looking at, and we can attach it with our other plots that we generated from our other sets of regressions to make sort of this like master plot. And what we're going to use this for is we want to find if there's any microbes that are also associated with a metabolite that's also associated with sleep. So this is how we would go about using this. For instance, maybe we're interested in Ruminococcus. We can see that it's associated with maltose, cysteine S sulfate, SAH, and 2-piperidinone. Okay, and then maybe we're curious about SAH. So then we can look across this way. Great. And then we also note that Ruminococcus and SAH are both associated with minutes of sleep. This X, M, which stands for mediator, and Y will become more clear in the following slides. But basically what we're doing here is we can find all of these triplets like we've done here. So we can do that for any of these boxes. And we can make these triangles and then run another analysis called causal mediation analysis to try to estimate what is the effect of X on Y that goes through this mediator. Okay, so let's get a little bit of background on causal mediation analysis. This is what we're trying to estimate, is what is the effect of X going through M on Y. Essentially what we're doing here is we're running several different regression models, and then we are partitioning the effect of this microbe X on a sleep measurement Y into the direct and indirect effect, also called the average causal mediation effect. So we're trying to partition out how much of this effect is through this metabolite. And so if you look here, this is the total effect. So that would be the sleep measurement regressed on the gut microbes. And then we can look at the mediator regression model, which is the mediator regressed on the microbe. And then if we specify a third model where we control for the mediator, this would be the direct effect, because this is the effect of the bug on sleep after controlling for the mediator. So anything that's left after this, that is the effect of the bug on sleep through the mediator. So we can subtract this coefficient from this one where we controlled for the mediator. If you want more information, this is a nice sort of introductory tutorial on how causal mediation analysis works. Okay, before we move on though, let's back up a little bit. We're looking at cross-sectional data here. This is not like an experiment. It's not a randomized controlled trial, and we can't make causal claims. Also, how I've presented it here, this might look like there's a gut microbe that produces a metabolite that goes into the blood that directly affects sleep. There's probably more steps involved. For instance, maybe a microbial metabolite is produced and it interacts with the peripheral immune cell or some enteroendocrine cell. And then there's hormonal signaling. Maybe you detect the hormone or some proxy for the hormone in the blood plasma metabolites. And then, that could cross the blood-brain barrier and affect sleep regulatory circuits in the brain, or there could be other intermediate steps here. Another important thing too is that I've drawn these arrows in this direction, but this is really contingent on whether we are adequately controlling for confounding. There's things like stress, inflammation, diet, polypharmacy that could be affecting the gut microbiota, blood plasma metabolites, and sleep measurements. And so the results that we're getting from the causal mediation analysis will not be reliable if there is confounding that we're not accounting for. There could also be a situation where the sleep measurements are impacting the gut microbiota. But despite these limitations, we're going to proceed with our analysis because importantly, this is setting up associations that we will later test in vitro and in animal models. Okay, so here's one of our hits. Okay, so we have the 3 legs of the triangle. And first we have Adler-Crutzia regressed on restless-duration. This is significant. Then we have the metabolite. We have the restless duration regressed on the metabolite. That's also significant. And then we did the third arm of the triangle too, where we see significant association between the bug and the metabolite. So now that we have all 3 of these arms, which we got from our 3 sets of regression models, then we can run the causal mediation analysis to estimate the Qiime. And we find that the Qiime is significant and we're estimating that it mediates 33% of Adler-Crutzia's effect on restless duration. Okay, so despite the limitations of causal mediation analysis used in this case, it's still a really great way to generate hypotheses. So we were just looking at Adler-Crutzia, and we found that it had a mediated effect on restless duration through to piperidinone. So something that's interesting is Adler-Crutzia belongs to this family, Agarthaceae. And these members are known to modify plant polyphenols and alkaloids, specifically species within the Adler-Crutzia genre are known to, they're known to modify transresveratrol. to dihydroresveratrol. And they are also known to reduce this molecule, daidzein, which is a polyphenol from soy, and they can convert it to S-equal, which has anti-inflammatory properties. So it looks like this bug is involved in several, well, in these two different reducing, like plant polyphenol reducing reactions. And so other data that I've not shown here, Adlercreutz is also associated with piperine, which may be an upstream metabolite of 2-piperidinone, which is what we found in our hits. So maybe there's some sort of like reducing reaction that Adlercreutz is doing on piperine to affect 2-piperidinone, which then affects the restless-stration. So we can take this and look, we can test it in vitro and then look at the results. So that's just one example of like a relationship that we can pull out from this data set. This Sankey plot is a summary of all of the triangles that we tested with the causal mediation analysis that had a statistically significant average causal mediation analysis. So we have a handful of things that we can test in vitro and then possibly in animal models. So in the future, I've done these. So I've extracted these relationships from the Airveil data set. But to be more confident in these relationships, we can mine external data sets and see if there is consistency across the data sets. Airveil also has multiple time points for certain people. So eventually I would like to look at how individuals' gut microbes or metabolites or sleep change over time. And then there's also diet, medication, supplement questionnaire data in Airveil that I haven't dug into yet that could complement the analyses that I've already done. And then we can take those findings, do some in vitro tests. I briefly talked about the one with Adlercreutz and 2-piperidinone, but we could test the other associations that we found too that were in that Sankey plot in the previous slide. And then also test some of these findings in mouse models by either feeding mice a certain microbe that we think affects sleep, seeing if it changes its sleep phenotype, or a metabolite, or both. So just wanted to thank the Gibbons Lab and everyone here. And yeah, thanks for listening in on my talk today, and happy to take questions. Let's see. Okay. Aha. Okay. So someone writes, are you sure that the mediation is running microbe to metabolite to sleep instead of metabolite to microbe to sleep? Is there a way to establish which direction is more likely if you don't know a priori? That is a great question. Thanks for asking. So let me share screen again. We have a little bit of, we have a little bit of a priori knowledge since we know that gut micrometabolites can enter the systemic circulation. But another way that if we didn't have any of that knowledge whatsoever, one way that we've approached this is we run the mediation analysis in both directions and then use a statistical test to see which direction is a larger effect. So I've run a gut microbe with the blood plasma metabolite on sleep and seen the effect of the gut microbe through metabolite on sleep. And then I've actually also run the reverse, where a blood plasma metabolite affects sleep through a gut microbe. And so I haven't shown that data here, but we can take this chart and then partition it into, okay, which direction is stronger? And so we could have like, if the forward direction is stronger, you would have like Blautia on cysteine S sulfate on sleep. And then we could have another plot that shows for the metabolite microbe sleep direction, if that direction's stronger. Okay. Somebody else writes, which prebiotics and or probiotics are found to improve sleep? So these studies are generally looking at, well, they're exclusively looking at lactic acid bacterial probiotics, and the specific strains, I can't remember the strains off the top of my head, but they're different lactobacillus. And then there are several that have, I think maybe like streptococcus thrown in there. Some are individual strains, some are cocktails of several different strains. And then as far as prebiotics, some of those prebiotics, they're like plant oligosaccharides. There was one study I read that gave the treatment group some like traditional Chinese medicine plant granules from a variety of different plants. There was another prebiotic study looking at milk thistle extract as a prebiotic. So yeah, it varies. Yeah. What is your perspective? Okay, so this is the next question. What is your perspective regarding the age of the patients in the cohort? Will older patients have different sleep patterns? That's a great question because sleep quality and architecture does decline with age, even in disease-free individuals. In terms of this cohort is primarily middle-aged individuals who are generally healthy. And then in terms of my data analysis, age was included as a covariate in all of the regression models. So the effects that I've reported here are independent of age, or you could also say the effects here are after controlling for age. But I think, yeah, even outside of this cohort, if you look in the literature, older patients do have different sleep. Oh, there's more. Wow. Okay. Somebody asks, would it be a good idea to test the directionality of the mediation with time offset measurements? Would it be a good idea to test the directionality of the mediation with time offset measurements? So if I'm understanding this question correctly, I think you're maybe referring to the fact that I'm applying this mediation analysis to cross-sectional data. And would it be a good idea to apply it to maybe time-resolved variables? Certainly, yes. In fact, the causal when you can make causal claims is if I, for instance, use this analysis in the probiotic paper that I showed earlier, which, where the heck is it? So for instance, here, they looked at, they gave people a probiotic and then the outcome variable was the PSQI score, but then they also looked at gut microbes and blood plasma metabolites. So what they could have done here with their data analysis is you could make that triangle like we did, where there's probiotic and it has an effect on sleep. And then you could, for your mediator, you could look at, a gut microbe or this blood serum metabolite, and you could say whether the probiotic had an effect on sleep through a certain gut microbe or a certain blood metabolite, and what was that effect size. There's also ways that you can include multiple mediators. That's a little bit more complex and outside of the scope of this. Okay, somebody else asks, have you looked at the effects of the covariates independently? Are there sex differences? So yes. Have I looked at the effects of the covariates on sleep independently? Yes. Sex does have an impact, as does BMI in this cohort, as well as several of the activity measurements. And actually, to account for some of the collinearity across the covariates, I was actually able to reduce or get rid of some of the activity features because they were really co-correlated with each other. But yes. Okay, since metabolite leads to disruption in sleep, is vice versa also possible? And how do we take that into account? That since metabolite leads to disruption in sleep, is vice versa also possible? Yes, I would say that's definitely possible. The communication between the gut microbiome and brain it's a crosstalk, so it's bidirectional. And kind of like Alyssa alluded to earlier today, not only do microbial metabolites influence the brain can also send signals to the gut, which influence the microbes among other ways. And then in terms of how we take that into account, I think this question is similar to the other question in perhaps asking, does a blood plasma metabolite affect a microbe which affects sleep? And we can account for that by defining our mediation models in this opposite direction. And then the effect that we get when we go from metabolite to gut microbe to sleep, we can compare that estimate to the microbe metabolite sleep and then see which direction is stronger. Okay, have people looked at diet or dietary questionnaires in addition to metabolites? People looked at, yeah, I think one of the major issues with the dietary questionnaires is that they're really noisy data, and there's, reporting biases too. People might not report faithfully, like exactly what they eat. And then there's also the issue with, I think, trying to align the questionnaire data with the metabolite data time-wise or the microbiome data. I think tomorrow, our lab has developed a computational tool to estimate dietary intake from your poop directly. So that way we have kind of an estimate of what you ate exactly paired with the 16S sequencing data. So that's pretty cool. And then another challenge of like the diet data, at least in this cohort, is that it's really unstructured. So there's a lot of data cleaning to do to actually extract any usable data. Okay. Yeah, great. And if that's all the questions, then I will hand it off to Sean. Thanks so much for participating today, everyone.
Great. Thanks, Jacob, for the great talk. That wraps it up for day one of the course this year. Thanks again to our sponsors, Applied Microbiology International, the Environmental Health and Microbiome Research Center, or the Embrace Center at the University of Washington, and Science in Seattle. Special thank you to the team today. Our instructor, Alyssa Eastonon, did an incredible job. She added all of this covariate adjustment and differential abundance analysis as a new component of this year's course, which I think was a wonderful addition to the course this year. And Jacob Cavon for giving this great talk on how do we actually use these tools to understand the gut-brain axis in a real-world example. So ISB is a nonprofit research organization. You can learn more about us at isbscience.org. To recap what you learned today, right, you kind of went from raw sequencing data input for 16S amplicon sequencing. You processed those data, you turned it into abundance matrices that could be analyzed and manipulated. You did sequence alignments and phylogenies, turned all these things into visualizations, and then learned how to run statistical tests to understand whether there are differences between your cases and your controls, all focused on Parkinson's disease, which is an important gut-brain axis condition. So tomorrow, for day 2 of the course, we're going to continue down along this Parkinson's disease line. We're going to use the same data set you processed today, and we're going to leverage a modeling platform that was created in our group by Dr. Christian Diener, who's now a professor at the Medical University of Graz in Austria. And our instructor tomorrow, Dr. Alex Carr, will be walking us through how to use MyCom to analyze the functional outputs of microbiomes. We will kick things off at 9 a.m. Pacific time or Seattle time tomorrow morning, same as today. We hope to see you all there. Thanks again for joining us today, and have a great rest of your day, and we'll see you tomorrow.